So this session is talking about the mitosis and meiosis, which is about the basic mechanism of sexual reproduction, documentary meiosis. Biology is the only science where multiplication and division, they have the same meaning. They mean the increased number. So mitosis is the process by which somatic cells, they undergo division and they reproduce new daughter cells. These daughter cells, they are identical to the mother cell. They have the same genetic information and they conserve the genetic information from one generation to another. How would the cells need to reproduce? Why do they need to reproduce? They need to reproduce in order to increase the number of cells by the increase of the cycle and to renew the body tissues which are injured or dead, or for example, they are old. You have to know that the life cycle includes interphase with the mitosis. Interphase is divided into three checkpoints: G1, S, G2. G1 is called the gap 1. S is the synthesis phase. G2 is the gap 2 phase. Synthesis phase where duplication or the replication of DNA takes place. Mitosis is divided into four main phases: prophase, metaphase, anaphase, telophase. So DNA synthesis or DNA replication occurs during the S phase of the interphase. G1, gap 1, S, DNA synthesis or replication. G2, which is gap 2. Then we have mitosis, and finally we have cytokinesis where the cell is going to divide into two. So you have to know that at the interphase we have chromosomes, they are not well formed, we have chromatin. Okay. Then during the prophase we have the nuclear membrane. This integrates. You have to study these events. You have to know what's happening in every single stage or checkpoint during the mitosis. You have to know that the nuclear membrane disintegrates. Mitotic spindle fibers, the one in green, start to form. Centrosomes, they change into asters. The chromatin condense so that you have individual chromosomes, each are made up of 2 chromatins each. So these are the phases. Nuclear membrane disintegrate or disappear. The mitotic spindle fibers, they are formed. We have the centrosomes, they change into aster, and chromatin condense, so that we have individual chromosomes made up of 2 chromatids each. Prophase, then we have the second phase is called metaphase. Metaphase, where the chromosomes, they line in the middle of the cell, forming what's known by equatorial plate. That is the role of colchicine. Colchicine, they are, for example, it's used to stop or to arrest the cell at metaphase. Then we have the polar ascension, which is special for anaphase, where the sister chromatids are pulled apart by the spindle fiber to the opposite pole of the cell. We have each set of chromosomes, each group, they are formed of chromatids, 1 chromatid each, and they move to the opposite pole of the cell. This is known by polar ascension. At the end of the anaphase, the result will be 2 identical groups of chromosomes, each made up 1 chromatid at the opposite pole of the cell. At the end of mitosis, we have telophase and the cytokinesis. Cytokinesis and the cleavage. The cleavage in the cell, whenever it cleaves and divides into 2, this cleavage is called the cleavage furrow, where the cell starts to divide to give 2 daughter cells. The chromosomes form 2 identical groups at the opposite pole of the cell. These chromosomes will decondense back to give the chromatin form. Nuclear membrane will reform, reappear, spindle fibers disintegrate, and the asters return to the centrosome state. Cell membrane constrict. This constriction is referred to as cleavage furrow to divide into 2 daughter cells that are identical to the mother cell. You have to keep in mind these notes that whenever the cell constricts, this cleavage of the cell into 2 daughter cells is referred to as cytokinesis. And we have to know that the 2 daughter cells obtained, they are identical to the mother cell, and they have the same genetic information, and both of them, they are typhoid. This is everything related to mitosis. So you have to know mitosis, what happens in this stage. Note that in the plant cells, there is no asters, and there is no cleavage at the end of the telophase due to the rigid cellulose wall. You have to know that plants, we have a wall that's called rigid cellulose wall. A rigid means that it's very strong or thick wall that is not able to constrict and divide, so there is no constriction. So new cellulose wall will form around the cell in the middle, leading to the formation of two daughter cells. So there is no cleavage or constriction. This is at the end of interphase you have in the chromatin form, meiosis. We said first, mitosis, they are the stages or they are the set of events that occur for somatic cell, to diploid cell, to give two daughter cells that are identical to the mother cell and having the same genetic information. Now, meiosis is the process by which the mother cell of gametes, diploid, to N, produces gametes, haploid. This occurs mainly in the sex cell, the gametes from the ovary. So the mother cell of gametes go through an interphase in preparation for meiosis. You have to know that we have two divisions. You have meiosis 1 and we have meiosis 2. Meiosis 1 is also known as reductional division, whereas meiosis 2 it's referred to as equational division. Why they are called equational? Why they are called reductional? And let's see. First, meiosis 1, prophase 1. As you see here, the chromosomes, they form one over the other. They form like this, okay? The two chromosomes, they form one over the other. This is called the tetrads, formation of tetrads. The chromosomes, they move one over the other. So this is called the pair up of the chromosomes. So the nuclear membrane for phase 1 disintegrates or disappears. Spindle fiber start to form. Centrosome change it into aster. Chromatin condense so that individual chromosomes each might have 2 counted each. As you see here, this 1, 2, 3, and 4 steps, they are very similar to that of mitosis. The only difference is that the homologous chromosome, they synapse. At the meaning of synapse, they pair up. They pair up. For example, they pair up forming what's known by tetrads. Tetrads, we have 1, 2, 3, 4. So they are called tetrads. This increases the chance of the crossing over between the son-sister chromosomes. Meiosis 1, prophase 1, here we have metaphase 1. Metaphase 1 is the same. They line up at the equatorial plate, forming what's known by equatorial plate or the equator of the cell. But here, not chromosome, they line up. Here, the tetrads. Tetrads that are formed during the prophase. They line up at the middle of the cell, forming what's known by equatorial plate. Anaphase, we have chromosomes, they move to the opposite pole, not the sister chromosomes. So spindle fiber will pull the homologous chromosomes to each tetrad apart. Each chromosome will move to the opposite pole of the cell, and this is known by polar ascension. Late anaphase, we will have 2 groups of chromosomes, each made up of 2 chromatids at the opposite pole of the cell. Moving to the last phase of meiosis 1, telophase 1. Telophase 1, the result will be 2 daughter cells. So during telophase 1, cell membrane constrict. We have constriction of the cell membrane, forming the cleavage furrow. New daughter cells obtained where each carries 1 copy of each chromosome. They are made of 2 chromatids each. Obtained cells here, they are called haploid. They are 1N. So the number of chromosomes is reduced. That's why meiosis 1 is called reductional, because the number of chromosomes is reduced into half. So daughter cells, they are different from each other and they are different from the mother cell. Moving now to meiosis 1, also telophase 1. The chromosome will form 2 groups, as we said. Of the cell, the spindle fiber disintegrate and the aster return back to the centrosome state, the nuclear membrane before. Note here that there is no interphase between meiosis 1 and meiosis 2. The interphase occurs only before meiosis 1. So before meiosis 1, we have interphase, then meiosis 2, and after meiosis 1, immediately, we will have meiosis 2. Here we have 2 daughter cells, meiosis 2, prophase 2. The mitotic spindle fibers start to form. Centrosome, they change into aster. The nuclear membrane disintegrates. These events, they are greatly similar to that in mitosis. Metaphase 2, chromosomes, they line up at the equator of the cell, forming what's known by equatorial plate. Meiosis 2, anaphase 2, the sister chromatids, they are pulled by the spindle fibers to the opposite pole of the cell. They form. Each group is made up of one chromatid at the opposite pole of the cell. As you see here, we're looking at two daughter cells and not one daughter cell. And telophase 2 and cytokinesis, the result will be four daughter cells. These four daughter cells, they are different from the mother cell. The chromosomes form two groups at the opposite poles of the cell. These chromosomes will decondense back into the chromatid form. The nuclear membrane start to form around each set, and the spindle fibers will disintegrate, and the asters return back to the centrosome. Okay? You have to be careful to this. And you have to know, for example, if I give you a figure like this, you have two daughter cells, so it's which cell cycle or which cell division, it's mitosis. While if you have four daughter cells, so it refers to meiosis. This table is a very important table. This table is showing the difference between mitosis and meiosis. You have to know that mitosis occurs in somatic cells, every single cell except the gonads. The aim of mitosis is the growth, like the case of the zygote, or the renewal and the replacement of the cells or dead or old or injured. While the aim of meiosis is the production of haploid gametes, 1N gametes. Mitosis occurs in only one division, producing only two daughter cells, whereas meiosis, it includes two successive divisions, producing four daughter cells. Mitosis occurs to somatic cells in all the body organs, skin cell, muscle cell, heart cells, liver cells, pancreas cell, while meiosis occurs only the mother cell of gametes in the gonads. Okay, only for the gametes, sperm and ovum. Mitosis is made up of one division. It's preceded by interphase, whereas meiosis is made up of two divisions preceded by interphase. We have no pairing of homologous chromosomes, so there is no formation of peptides, while in meiosis, we have pairing of homologous chromosomes and we have formation of peptides. In mitosis, daughter cells obtain 2N, one chromatid per chromosome, while in meiosis, 1N, one chromatid per chromosome after the finishing of meiosis 2. You have to know that meiosis 2 is greatly similar in its events to that of mitosis. Okay?